Welcome to our program tonight, a Charlie Rose special edition. In the 10th episode of our brain series, we look at the disordered brain.
Today we're going to discuss these neurological diseases, and we're going to also see that a fundamental difference between neurology and psychiatry is that, by and large, we don't know very much about the anatomical underpinnings of most psychiatric disorders. We don't know the neural circuitry that's responsible for schizophrenia, bipolar disorder. We have remarkably good insight into the neural circuitry underlying most neurological disorders.
The 10th episode of the Charlie Rose Brain Series, underwritten by the Simons Foundation, coming up.
From our studios in New York, this is a special edition of Charlie Rose.
Tonight we continue our journey through the fascinating world of the human brain. When working properly, the brain performs sophisticated tasks smoothly and easily. It allows us to move, to speak, and to interact with our surroundings, requiring only minimal amount of effort. But when the brain is damaged, its true complexity is revealed. Our subject this evening is the neurological disorders. These include Parkinson's disease, stroke, Huntington's disease, and spinal cord injury. These conditions have taught us more about our brain than any other kind of brain disease. Through Parkinson's, we have learned about movement. Through stroke, we have learned about speech. And through spinal cord injuries, we have learned how thoughts give rise to actions. Neurological diseases have been a topic of research for centuries, but only recently have we developed effective treatments. This evening, we will meet a group of scientists who have developed ways to repair or bypass the disordered brain. John Donahue. His work allows paralyzed patients to move and communicate using only their thoughts and a machine called a brain-computer interface. He is a professor at Brown University and the co-founder of a company called Cybernetics. John Krakauer. His work explores how the brain controls movement and how movement is recovered following a stroke. He is an associate professor of neurology and neuroscience at Columbia. Nancy Bonini. She studies the genetic basis of neurological disease by performing experiments on fruit flies. She is a professor at the University of Pennsylvania and a Howard Hughes medical investigator. And joining me from Atlanta is Malin DeLong. He is an expert on Parkinson's disease and a pioneer in the growing field of deep brain stimulation. He is a professor of neurology at Emory University School of Medicine. And once again, my co-host is Dr. Eric Kandel. As you know, he is a Nobel laureate, a professor at Columbia University, and a Howard Hughes medical investigator. I am pleased once again to have him here to help me understand all about the brain. So welcome back.
Thank you. You're doing very well understanding the brain.
And what a journey it is. So tell me about today, the disordered brain.
Well, last time we discussed psychiatric disorders. Today we're going to discuss neurological disorders. These are both disorders of the brain. So one of the first things we want to explore is how do they differ from one another? What's the logic of neurological as compared to psychiatric disorders? Now, there are two fundamental differences. One in the nature of the symptoms, and two in anatomical location. In terms of symptoms, there is overlap. But from a simplified point of view, you could say that psychiatric disorders deal with enhancement, exaggerations of our everyday life. We all feel despondent periodically. We all feel hopeless and worthless when an experiment doesn't work. And this is an extension what you see in depressed people. The mirror image, euphoria, we experience when things go well, when we have a wonderful roundtable discussion. And we see a dramatic extension of that in manic disorders. And even schizophrenia, the hallucinations and delusions, we have examples of that in our dreams. By contrast, neurology differs in two ways in terms of symptomatology. First, we see fragmentations of symptoms. In Parkinson's disease, we see a dramatic slowing of movement. But in addition, we see the appearance of behaviors that you don't normally see otherwise. With a lesion of the parietal lobe, we see neglect of a whole side of the body. Just absolutely remarkable, a denial that it's there. But perhaps an even more fundamental distinction is anatomical. We know very little about the anatomical location of psychiatric disorders. what are the brain regions that are involved in schizophrenia and depression? We're just beginning to explore that. With neurology, we have an amazing understanding of the neurological underpinning of neurological diseases. And we've known this for years. When I was a medical student in the 1950s, clinical neurology was known as the medical discipline that could diagnose everything and treat nothing. This has changed dramatically. As we're going to learn in this program, there are major new treatments coming along in neurology that have revolutionized treatments in various neurological disorders: Parkinson's disease, stroke, even spinal cord transections. Now, one of the interesting things about it, this progress again has involved finding out more about the location of specific neurological diseases. It's location, location, location that counts in the brain. And the history of how we localize functions itself is so fascinating. It began in around 1860 with Paul Broca. Paul Broca was interested in disorders of language, the aphasias, and much of what we learned about the early localization of functions came from studies of language disorders. Broca encountered a very interesting patient one day. He had an aphasia, a language difficulty, which took the form of the fact that the patient could not express himself very satisfactorily in language. He could understand language perfectly well, but he couldn't make himself understood. This was not simply a paralysis of the vocal cords. He could hum perfectly well. Moreover, he couldn't write language. He just could not express himself in language. When he died...
But he could understand.
He could understand perfectly well. When he died and came to autopsy, Broca examined him and found a lesion in the front of the brain. He wanted to talk about this to his colleagues, so he had to give it a name, and in all modesty, he named it after himself, Broca's area. He then encountered 8 other patients that had a similar aphasia. They could not express themselves in language, but could understand perfectly well. When they died and came to autopsy, he found invariably they had a lesion in the front of the brain, and invariably the lesion was in the left side. And this caused him to realize one of the major insights we have in the biology of the brain. We speak, he said, with our left hemisphere. The left hemisphere is specialized for speech. This galvanized the scientific community. People began to look for other localizations of functions. And 10 years later, Fritz and Hitzig, 2 German investigators working on dogs, found that there is a systematic representation of body movements on the surface of the brain called the motor cortex, or the motor strip. If you stimulate one part of the motor strip, your face moves. You stimulate another, your arm moves. Stimulate another, the leg moves. So there's a systematic representation of your body movement on the surface of the brain. This is extraordinarily exciting. And a few years later, in about 1875, another giant came on the scene, Carl Wernicke, an extraordinary guy. A couple of years out of medical school, before he was 30 years old, he made a fantastic discovery. He found another language deficit, another aphasia, but this was a difficulty in understanding language, not in expressing it. So this patient could express language perfectly well, but he couldn't understand language. When he came to autopsy, Wernicke examined him and found a lesion not in the front of the brain, but in the back of the brain.
But the left hemisphere in the back.
In the left hemisphere, but in the back. So again, he had to name it so he could speak to other people about it, and he called this Wernicke's area.
Right.
And he said to himself, isn't it interesting? We're dealing with a complex function like language. This is not localized to a single region. This involves at least 2 regions, one for understanding language and one for expressing it, like a sensory area and a motor area. And when he looked to see where they were located, he realized that the Wernicke area, the area he had worked on, was at the back of the brain. This is where sensory information comes in. So when
understanding is where the sensory information comes in.
Exactly. So information from reading or hearing feeds in from the back of the brain into the auditory cortex and the visual cortex. These 2 areas converge onto Wernicke's areas and put that visual information and auditory information in a code for understanding language. He also knew there was a connecting pathway called the arcuate fasciculus that went from Wernicke's area to Broca's area. So the information goes from the sensory areas to Wernicke's area, from Wernicke's area through the arcuate fasciculus to Broca's area.
Now, what kind of connection is that?
This is a pathway, an anatomical pathway. Right. And that question that you raised made him realize a very profound thing. He said, I bet you one can get an aphasia, a language deficit, without damaging Broca's area or Wernicke's area by simply interrupting the pathway. You got it. You're a functioning neurologist. Interrupting the pathway between the two. Under those circumstances, the patient understands, the patient can speak, but there's no connection between the two. It's like an occasional presidential press conference. Information comes in, information comes out, but there need not be a connection between the two.
All right, so let me ask about this too. Where is the genetic understanding taking us?
Well, we have several areas in which we made terrific progress, okay? So number one, in addition to understanding the areas that are involved in speech, we have a very good understanding of the areas involved in the movement, okay? And Malin DeLong has discovered still another area important in the basal ganglia for Parkinson's disease. And he and Alan Benamid have collaborated together and used deep brain stimulation to produce dramatic improvements in Parkinson's disease. John Donahue has done something equally remarkable. You mentioned the fact that there are specific areas that are involved in movement, the motor strip. They fire in a characteristic way when you pick up a glass of water. He realized he could use that information to drive a computer. So if you want to pick up a glass, but you're paralyzed, so you can't do it, that information from your motor cortex can go onto a computer that can drive a robot to pick up that glass of water for you and bring it to your mouth.
This is extraordinary, and it must have enormous potential.
It has tremendous potential.
You know, if you can think, you can do.
Exactly. It also gives you additional confidence in the fact that we're reading the brain correctly. John Krakauer is on the verge of revolutionizing the treatment of stroke. He thinks the period of plasticity involved in stroke is greater. And the question that you originally raised, there is a lot of genetics that we now understand about these neurological disorders. And Nancy Bonini has taken these genes that are disordered in neurological diseases and put into flies a terrific experimental system where you can study what we call mechanism of pathogenesis. How does the disease come about? So we're going to have a marvelous evening together.
Okay, all that you wanted to know about neurological diseases, the disordered brain, both neurological and psychiatric. We go now to our panel. Let me begin with you, John. Tell me sort of a primer on these neurological disorders and how we approach them.
Right. So I think following on from what Eric said, the study of neurological diseases is important for two related reasons. One is that they help us gain insight into how the normal brain is put together. Because in everyday life, our behavior seems so seamless, so easy. It's not until we have an injury to the brain that we suddenly see how, in fact, this seamless behavior is made up of component pieces. And so we can gain insight into those components and how they're actually organized in the brain in neurological disease. And then the second related area is that by studying neurological diseases, we can get insight into treatment. So I'd like to sort of give two examples of the kind of insights we can gain from studying neurological patients. And I'm going to use this brain as an example. A big debate raged in the 19th century between two camps. One camp that thought that brain function was divided up into localized components, and another camp that thought the brain did everything holistically, right? And interestingly, that debate goes on today, but and the reason for this debate was because it all depended what kind of disease you decided to study. So, for example, if you get a stroke in this motor strip here, you develop a paralysis in the limbs on the opposite side of the body. But otherwise, you're okay. You think okay, your language is okay, it's just the limb on the opposite side of the body that is affected. So that was a very strong case for highly localized function. But there are other kinds of lesion that cause a much more diffuse, broad set of abnormalities, which made it very difficult to believe that all of those could be actually controlled by one region. So, for example, you can get something called neglect, especially when you affect the right side of the brain, and you get the inferior part of what's called the parietal lobule. In these patients, they have great difficulty orienting, attending, and acting to stimuli in one half of visual space or in on one half of their body, in this particular case, the left side. So, for example, if you touch them after this lesion here on their left side, you ask them where you're touching them, they'll say, my right. When you walk into the room from the left, they'll go, hello, and turn their head to the right. Okay? So profound cognitive problems caused by a fairly focal lesion. So in other words, in some cases, the processing is very local and you get a very focal effect, and that's why some people thought that it was all very localized. And in another case, you have a focal lesion that has diffuse effects because of a knock-on effects to other regions, and you get a more global abnormality. And so you can see, by continuing to study patients with imaging techniques that allow us to see this architecture, we can actually learn fundamental principles about how the brain is put together. So Parkinson's patients have a part of the syndrome is bradykinesia. They move slowly. So a colleague of mine, Pietro Mazzoni, asked a question that one tends not to ask. Why do Parkinson's patients move more slowly? And the converse question is, why do we move at the speed that we do? So I'd like to do a little experiment just to show that. Is if each of you reaches for your glass of water, you'll find that all of us pretty much reach at the same speed. So on the one hand, you voluntarily decided to reach for the glass, but on the other hand, you unconsciously picked a speed which was very similar to all of ours. And what we think might be happening is that they have a different balance between how much effort is required to reach the glass and how worthwhile it is to reach the glass. And there, unconsciously, the balance is moved towards it being too effortful to move fast to get it in the same time that we do.
Give us a sense of sort of what the history of what we have understood and how we've treated Parkinson's and where we are today.
James Parkinson's in 1817 described Parkinson's. It was the first description: slowness of movement, characteristic tremor, and the shuffling gait. In fact, he called it the shaking palsy because of the tremulousness and the slowness, palsy meaning weakness. He did not name it after himself. It was 150 years from that time, roughly, that the first really effective treatments were developed. Yeah. And ironically, these were not medical but surgical, neurosurgical procedures. Neurosurgeons, desperate to find treatment for patients with uncontrollable, excessive tremulousness, were able to identify, largely by trial and error, specific regions in parts of the deep brain circuits, and this involves the basal ganglia and a region called the thalamus. But in the 60s, there were a remarkable series of discoveries. The first was that the presence of a substance called dopamine within the brain, and particularly in the basal ganglia, and then in rapid succession, the evidence that it was depleted in brains of patients who were at autopsy with Parkinson's. These two discoveries led to the attempts to replace that substance by giving a drug called levodopa. Levodopa is a precursor of dopamine and taken up and absorbed and actually reaches the brain. And this was viewed as a cure. It was a matter of time, though, that the other foot dropped, so to speak, and began to realize that after the honeymoon period of a number of years, often patients would develop involuntary movements, we call them dyskinesias, or they would have what we call motor fluctuations, where the medicine would wear off abruptly. And these combination of motor fluctuations, sometimes failure to response, and the dyskinesias would be very disruptive of movement. And this called for really alternative approaches. During the 70s and 80s, there was a remarkable series of, I would say, studies and progress in understanding the motor system, the anatomy, and physiology. And one of the most important thing, I think, and relevant to Parkinson's was the understanding that the basal ganglia, which were depleted of dopamine, were doing something more than just providing some substrate for movement. In fact, it was commonly believed at that time that the basal ganglia were primarily movement because of the association with disturbances of movement and basal ganglia dysfunction. We were able to show that, in fact, it was a restricted part of the basal ganglia that were really involved with movement, and that other regions were involved with higher function. The key to finding understanding the mechanism of this was actually to lesion, to destroy a small part of the motor circuit within the basal ganglia. And this is a region called the subthalamic nucleus. The details are not so important, but
this
to
is Malin's
realize that
discovery.
this
It was just a fantastic advance to realize that this area, which is by and large neglected in the basal ganglia, had a central role in the control of movement and in Parkinson's disease. And that's what led you really to start thinking about using deep brain stimulation for that.
We were, at first, and I think others, most concerned not to lesion the subthalamic nucleus. So it was really Alan Benabid's discovery of the deep brain stimulation when he applied it to the subthalamic nucleus that was so important in the development of this new approach. By the 1990s, deep brain stimulation had virtually replaced all lesioning approaches to treating Parkinson's disease. This was the great advantage of deep brain stimulation, was the non-destructive, reversible, and adjustable nature of this treatment. I'm going to show you one of our first or earliest patients. This was our 17th patient who had received deep brain stimulation, and this was in 1997, and we'll let this roll.
The disease had gotten so bad that sometimes her muscles froze completely, making her face almost expressionless, and her legs almost useless. The woman who once was always on the go could barely move, confined to a wheelchair.
Do you ever wake up in the morning and forget that you have Parkinson's for a moment or two?
Not now. Not recently. Because my every waking hour is terrifying.
But on the morning of the surgery, Sybil was confident, even as nurses fitted her with a head frame to help guide surgeons during the operation. And as they wheeled her to the x-ray, she was in good spirits. Deep brain stimulation comes down to one thing: location. To help pinpoint the region of Sybil's brain where he would be working, neurosurgeon Roy Baquet used magnetic resonance imaging. Finding the target area holds the key to Sybil's future. If the stimulator is implanted correctly, she may finally be free of the tremor and pain that have haunted her for years. But if the placement is off, the results could be devastating. Finally, the doctors think they have placed the electrode in the exact location that will help Sybil. When they turn on the stimulator, she suddenly shows an astonishing range of motion in her legs and hands.
Pick a leg up at all? There you go. Up and down. Drop it down now. Now pick it up. Great. And she couldn't do that before. So, how about the hand? Can you open and close your hand for me now? Good. Okay.
And when Dr. Vitek turns off the stimulation, Sybil's tremor and stiffness return.
I turned off the stimulator. Don't be worried. Okay. Can you move your ankle up and down? It's hard to do for her.
It was only a month after her second surgery when we caught up with Sybil again.
When I talked to you before you had any surgery, one of your biggest problems was that you couldn't turn over in bed.
I was really concerned about that. But now, I roll back and forth. You can't stop me now.
Now, the woman who never wanted to slow down doesn't have to.
Show me what can you do now that you couldn't do before?
Oh,
Good heavens, look at that! Look at her! Look at that!
Sybil and Alvin had hoped for a lot from the surgery, but the outcome was better than they could have imagined.
Underline what that tape shows us.
I think the most important thing is that it shows the clinical benefits and it shows a profound effect on quality of life. Sybil, this simple video shows that better than any.
It also shows how scientific the treatment had become, because they first introduced the electrode for deep brain stimulation on one side of the brain, and it stopped the tremor on one side of the body.
Right.
And then, in order to control the other side, they second, as an afterthought, put in the electrode on the other side and stopped it on the second side of the body. So the patient now was free of tremor, and she moves from being essentially paralyzed to jumping and dancing around with the family. An extraordinary improvement.
Does it work on everybody? And what have we learned about it?
Yeah, that's a very important point. This does not work for everybody, and it's not a cure. It's a completely symptomatic kind of treatment. And if the battery should fail or the wires become disconnected, can happen, but not common. the benefit is lost almost immediately.
Let me turn to Nancy now and talk a bit about genes and how they're involved in the diseases we're talking about.
Right. So another approach to get towards these diseases is to take a very basic research approach and use animal models. And an example of an animal model is actually Drosophila, which is the fruit fly. It has really powerful genetic approaches. So for many of these human neurodegenerative diseases, in fact, for many human diseases, there are gene inherited forms as well as the more spontaneous so-called sporadic forms. But we have ways in the familial situation where a clear gene is involved to get that gene, and then we can study that gene. So for Parkinson's disease, for example, one of the key genes that was identified is a gene called alpha-synuclein. So alpha-synuclein is mutated in rare inherited forms of Parkinson's disease. Well, it turns out that alpha-synuclein also accumulates abnormally in the brain of all Parkinson's disease patients. This has allowed the idea that even though you find these genes through these rare inherited forms, studying of that gene, that's right, they're going to tell you about the disease in general, even though most of the disease is sporadic. So with a gene, you can then study the ways in which that gene may cause disease, and that's called studying the pathogenesis. And animal models are really key experimental tools in this, and there's many different types of animal models that you've actually touched on in this series, such as the nematode, C. elegans, or the mouse. And I'm going to talk about flies. That's the system that we use, the basic fruit fly. So the fruit fly was really used and developed as an experimental organism by Thomas Hunt Morgan at Columbia University, and he was interested in basic aspects of chromosomes and heredity. Later, Seymour Benzer, he opened up the world of the brain in the fly, and he focused on genes that were involved in behavior. And just like Mei-Lung talked about, there's regions of the brain that are isolated, but they also connect to other regions of the brain. Genes don't work on their own, but genes work in a network with other genes, and these are called gene pathways. So in fact, we now know that for many processes, not only genes, but entire gene pathways are shared between flies and humans. So that means that we can then take that gene and study it and ask how it may be functioning and hope to learn not only about flies, but really what we're interested in. We're interested in people, in humans, right? And so the fly had been classically used to approach development, and then more recently it's become very popular for behavior. And we decided, well, why not try with human disease? So the idea would be that whereas these diseases in humans can take decades for their onset, in flies, we can give a fly a phenocopy, or what it looks like to have Parkinson's disease, in days to weeks. So we can grow many flies, and we can speed up the whole process in order to really study that mechanism. So on the next slide shows sort of a section through the fly brain where we're studying some aspect of Parkinson's disease. So an idea is when we have the gene, like alpha-synuclein, we can put that gene into flies and then ask, can we get an effect in the flies that looks like Parkinson's disease? Well, as Mei-Lung talked about, dopamine is really important in Parkinson's disease, and so we can express that alpha-synuclein Parkinson's disease gene in the dopaminergic neurons. So the top panel shows clusters of dopaminergic neurons in the fly brain normally. And what happens when we put in alpha-synuclein is seen in the panel. So we see fewer of those cells staining for this marker for dopaminergic neurons. So if we express alpha-synuclein in those cells, they become compromised and sick. And some people, you can even see movement disorders in these flies. So in fact, by expressing this gene that's associated with human Parkinson's disease, we can get effects in the fly that look like core effects of Parkinson's disease. So I'd like to show another example that's showing an effect on climbing behavior. So this is using a gene in the fly that's involved in ALS. So ALS is another really
amyotrophic lateral sclerosis.
Yes, it's another
Lou Gehrig's disease.
Yeah, Lou Gehrig's disease. It's another very debilitating disease, and it leads to loss of voluntary movement control and eventually paralysis.
This is what Tony Judd suffers from.
Right.
Right. So if we look at the behavior of normal flies will climb upwards. They have this robust negative geotactic response. But the flies that are defective in this human ALS gene, these flies are alive and they're moving, but they can no longer climb. So this is a remarkable example of sort of a fundamental phenocopy, or we're mimicking the disease in the fly situation. So of course in the real laboratory, we take a very comprehensive approach to this. So we look at many, many different aspects of what it looks like in the fly to express one of these genes. And we're asking how many features look similar to the human situation so that we can say how well does the fly show the fundamental features of that disease. And so in this situation, we're sort of using the power of conserved pathways and conserved genes in order to approach the really complicated problem of human disease. Let
me now move to John Donahue and talk about brain-computer interface, and give me a sense of a history of that, too, and where we are today.
Sure, Charlie. What I'd like to do is go back to the idea of localization in brain circuits, and not the brain circuit for language, but the one for arm movement, and how we've taken the fundamental discoveries in that area and turned it into the ability to help people who are paralyzed move again. So first, I'd just like to show that there are what goes wrong in paralysis. So basically, in this slide, it shows there are two kinds of major roots of paralysis. One is that the central motor pathways are disrupted, and the other one is that the peripheral motor pathways are disrupted. And in both of those cases, the brain is intact and functioning, but its connection to the outside world is broken. And what we're trying to do is, would be interested in doing, is reconnecting the brain to the outside world. We can't fix those pathways. We don't know how to fix them yet. But instead of using a biological repair, we could use a physical repair. That is truly wires and fiber optic connections. So to do that, we relied on some very fundamental knowledge. One is the idea of localization. And Eric brought up the idea that there is this region, the motor cortex, and there's a region near the top of your head about the size of a quarter that controls your arm. And that we've known since about 1870. This was a great area of discovery. But interestingly, what the neurons in that area did was unknown until about the 1960s, when we started putting microelectrodes in the brain. And Ed Everts pioneered this technology of putting a microelectrode in the brain and listening in on a single neuron and asking what it did. And if you play the video, I think I have a single neuron, you can hear the popping.
It's very exciting.
So this is the true, so we could hear the crackling noise. So this is the true romance of the neurophysiologist, is our job as neurophysiologists to try and make sense of that. So you're actually listening to a cell that's related to movement. And those little popping noises are called action potentials, or spikes. And that's the code of the brain. And neurophysiologists try to decode the brain and make sense of movement. And from studying behaving animals, we learned that the motor cortex, arm area, is very interested in producing commands of when to move and which way to move, left or right. So we had some insight from these years of experiments. For about the next 2 decades, we explored those areas, and we learned that really this information was encoded not in a single neuron, but in populations, much like the information in a symphony is not encoded by the second violinist, but you have to listen to the whole orchestra. Right. And what was seriously lacking was the ability to try and capture many cells at once, like listening to the whole symphony. So we developed an electrode array. That is, instead of one, a tiny implant of many electrodes. That's shown on this slide. It's actually something about the size of a baby aspirin. It's implanted permanently in the arm area of the motor cortex, and then it comes outside, and the signals are brought to the outside in its current version. So it's very tiny, just not even the size of a penny. At that point, we said, well, here we have technology that allows us to peer into the brain. We have an understanding, at least a primitive understanding, of what that part of the brain, the arm area of the motor cortex, is trying to do. We could, in fact, take people who are paralyzed, that is, able to think about movement but not able to move, and connect their brain back up to the outside world. So I guess what could be seen as a rather bold step, but we sought FDA approval, and we were approved by the FDA to study a small number of severely paralyzed people, tetraplegics, that can't move their arms and legs. And we implanted 5 people so far in this small trial. And I think just by illustration, I'll show you one of the first patients. His name is Matt Nagel. He's playing a video game here. He's supposed to hit those things those are treasure chests and avoid the squares, which are actually goblins. It's a video game called He-Man. So you'd say he was playing that video pretty well, but not great. But in fact, Matthew was injured in his cervical spinal cord. It completely disconnected his brain from his entire body. He has the chip in his arm area of motor cortex, and merely by thinking about moving, he's actually making that cursor move.
I just think this is stunning.
Amazing.
I mean, that you can.
Amazing.
But again, it's a true example of taking the information from basic science. We had all the pieces, and we just put it together so that we could reconnect the body to the outside world.
And what are the possibilities of this?
Well, you'll hear more about the possibilities. I think we want to give a couple of examples that I think take us. So we've sort of stepwise gone through this. So one step is, can you control something physical? So we thought of something physical, a mechanical hand. So Matthew can't control his own hand, so we brought a mechanical hand.
Right.
And here we're seeing a video of Matthew thinking about opening and closing a hand, and there he is opening and closing his hand. It had a very profound effect on him because it was the first time in years that he had actually moved anything physically himself, but he's doing it entirely with his mind. Then to extrapolate that a bit further, we asked, well, could you control something practical like a wheelchair? This is actually a lady who has what's called a brainstem stroke. It's a little bit above the spinal cord. She's not only unable to move, she can't speak. But what she's doing here is using BrainGate, this chip, to control that wheelchair. So it's wirelessly beamed over to the wheelchair, and actually the control system is that little computer mounted on the wheelchair. And I had asked her to drive the wheelchair over to the door, and she had practiced for about 5 minutes before this. And here she is sort of not very gracefully, but driving it over towards the door, and you'll see it progress up over to the door. And of course, you know, the control isn't good enough. We wouldn't have her in the wheelchair. We just had her do it remotely. So at this point, we've shown in each of several different disorders, that includes spinal cord injury, in stroke, and even in 2 ALS patients, that's possible to decode the brain, sense the signals, decode the brain, and connect them to devices. But these are all demonstrations. We're not there yet where these are available for everyday use. But Matthew actually recorded some of his comments about his views on the technology, and I think I think this video will just, he can narrate it and tell you what he thought of the technology.
I envision this giving me more independence as far as changing the television, opening my shades, turning on my lights, which right now the nurses have to come down and do for me. So it will give me a sense of independence, not as much as I'd like, but it would give me some independence, and for me that'd be wonderful.
What is also so wonderful about this is not only the clinical effect, which is so dramatic, but it really shows that John has learned how to read the brain, how to use the information from the brain to drive a device. So that itself is a major insight, that you can make sense out of the action potential sequences in the brain. And it really brings us to John's aspirations here, John Krakauer, of what you hope to do with stroke. Why don't you outline for us your thinking about that?
Well, right. So I think it follows very nicely, because the very same plasticity in these extreme cases where you're disconnected, where you're going to have to bypass the severed pathways and control an external device with this plasticity. Well, in patients who have stroke, for example, where they still have some residual movement left, the question you want to ask is a similar one. Can you somehow augment their plasticity and sort of enhance their recovery? And so there are a number of points to make about that. Most patients after injury that are less extreme than what we just saw recover to some degree. And they recover most of the time within the first 3 months after stroke, but not completely. And we know from animal models that we were talking about before that this seems to be because of a time window of plasticity that finishes after about 3 months in humans. It's about 4 weeks in rodent models. So what we want to do is, can we use technology to try and take advantage of this period of plasticity in patients after stroke. So there are two kinds of technology that are being used increasingly now. So here is a Rio Go robot. This is basically a robotic device that attaches to the affected limb of a patient after stroke. And what this robot can do is it can give varying degrees of assistance to the patient so that when they start to make a movement, the robot can give them 3D assistance. And as they get better, as they begin to understand the kind of movement required of them, you can begin to take away the robot's contribution. So the idea would be to take patients early after stroke and with the use of a robot which can give you hundreds, if not thousands of repetitions, and we know that large amounts of practice are required to actually interact with this plasticity to get recovery. So what we need is some sort of device that doesn't get tired and can be programmed to give you hundreds of thousands of repetitions and do it early. And these are what these robots can do. The second kind is to do something non-invasive. Unlike we just heard from John, in patients who are less severe, we don't necessarily think we have to put electrodes into the brain itself. We feel like we can actually stimulate motor cortex externally with either electrical or magnetic stimulation. So here in this slide you can see this figure-8 coil, and what you do is you put current through that coil, which then generates magnetic fields which are at right angles to the surface of the brain, and those magnetic fields induce current in neurons and stimulate a motor cortex. The other one, instead of using magnetic fields, you can use essentially a 12-volt battery over the head, and that also can actually increase excitability of motor cortex. And these are two ways to augment activity and plasticity over targeted areas of the brain, and a lot of studies now are showing that these can increase and improve performance. So the dream would be, the cocktail would be to take a patient early after stroke in this window of plasticity, hook them up to a robot and stimulate their brain at the same time and potentially give them some sort of drug, and with this cocktail ramp up the amount of recovery that could occur in these patients early after stroke.
Let me go back to Malin. This notion of where you see the future of the kind of research you're doing with deep brain stimulation.
As you know, deep brain stimulation is now being exported, if you will, from the Parkinson's tremor field to other movement disorders, has been successfully for dystonia and other disorders characterized by excessive movement. But more strikingly, and I think remarkably, for disorders that really are psychiatric in nature, such as obsessive compulsive disorder, depression, and a Tourette syndrome, which is a blend of movement and psychiatric disturbance. So I think the theme here is that we stimulate the motor circuit for movement disorders, we stimulate this emotional reward circuit for treating these psychiatric disorders, and it seems to be more circuit specific than disease specific. So we use the same targets, the same stimulation parameters for all of these conditions, granted that this is fairly large scale kind of stimulation.
There's an interesting sociological point here, Malin, and that is that Helen Mayberg, whom you had in this program recently, introduced deep brain stimulation for depression, stimulating this area 25 that she had found hyperactive. And here is a psychiatric illness that was treated successfully by a neurologist. And why is this so? Why weren't psychiatrists doing this? And that is the culture. The two fields are different. Yep. Neurologists intrinsically, from Broca and Wernicke on, have thought of anatomy, 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 location, 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 and psychiatrists have not thought in anatomical terms until just recently.
The main thing, Charlie, I would say about this technique is that it really has given renewed hope for patients with psychiatric and neurologic disorders.
The four people here have shown that in relatively short period of time, 1 or 2 generations, neurology has moved from being a field in which, with the exception of epilepsy, nothing was really treated effectively, to a point where it's really making major strides forward in treatment. I think this is a major, major advance.
So pick up in terms of... Right.
So you might ask, so I was talking about simple systems, using the fly and recreating a human disease in the fly, and you might ask, well, what's the point of that? Well, the point is that you can then use the very powerful genetic tools that are available in the fly to define genes and gene pathways and even drug compounds that can interfere with that. So, for example, in Parkinson's disease, alpha-synuclein actually accumulates abnormally. So it looks like the protein undergoes an abnormal folding process, clumping in the neurons. And this feature of abnormal clumping of disease proteins is actually a feature of many different human neurodegenerative diseases. So, for example, in Alzheimer's disease, there are plaques and tangles. So this is sort of a common feature that the proteins seem to build up and they clump abnormally in the cells, and this is probably leading towards the toxicity, the pathogenicity. So we have, and flies have, very conserved pathways that are critical to help proteins take on their normal shape, so to prevent this kind of clumping. And these are called molecular chaperone pathways, so these are helper pathways that can help proteins fold properly. So we considered if alpha-synuclein is causing toxicity, potentially by clumping abnormally, what would happen if we gave the flies more of these helper proteins? So recall in the top panel, the fly has a set of dopaminergic neurons, and in the middle panel, those become compromised so that there are fewer cells staining if we express alpha-synuclein in the dopaminergic neurons. But now in the bottom panel, we can see what happens when we add this helper protein, this really great molecular chaperone called HSP70. We, in fact, restored the system back to normal. We no longer see the compromise of these dopaminergic neurons. And I should emphasize that HSP70, this terrific chaperone, works not only in this situation, but it works in other fly neurodegenerative disease models, of which there are many now, and it also works in mouse models. So this is one example of where we sort of thought about what was happening, we stepped back from that, and we made a really good guess of what might be involved in the process.
John, let me finally come back to you and tell me where the frontier is in terms of brain interface.
Sure. Well, I think
brain-computer interface.
Well, it's not just brain-computer interfaces. I think that the future is to connect. First, I think for people who are unable to move and for whom we can't restore their full function, to connect them to external devices that are useful, that can help them in their everyday life. What many people don't appreciate is how devastating paralysis is, especially the tetraplegia that we saw. They can't do anything. So imagine if now you could control a very dexterous robotic arm that could get you a drink of water, because you can't drink without someone bringing it to you. So this shows now it's not a paralyzed person, but it's an able-bodied engineer. My colleague Jorn Vogel and his one of his co-workers, Patrick von de Smet, and they're showing this very sophisticated robot from the German Space Agency that he's controlling now with a real computer mouse with his actual arm underneath the table. But it's showing now, imagine a person could use their brain to control this and actually take a drink of water using this robotic arm under brain control. So we're actually underway doing this, asking our participants to try to accomplish this using a robotic limb. It will be slower than normal. It won't be as dexterous as normal, but we are very encouraged that we can have people once again interacting with their environment. But better than that, I think the next step for us is actually rewiring the brain back to the muscles with physical components. Yeah. It is possible to put stimulators into the muscles, bring the brain signals to the muscles, and so therefore someone thinks and moves. So our ultimate dream, which is very far away, I'm not sure any of us at the table will actually ever see this, would be that we'd be sitting here one day and one person would say, oh yes, I had a spinal cord injury, I've been rewired, I have several brain gates, I have a bunch of wires, I have stimulators implanted, but I can move just like you or me, and you can't distinguish me, and we'll go play tennis afterwards, you know. But of course, this is you know, very far out, but we're coming. We've actually done in a simulation with a patient the ability to make simple movements of their own arm, and this looks quite feasible. So I think you know, the future is very bright for a physical repair of the nervous system.
Right. What I always like to do at the conclusion of these conversations is to ask the one question: is that what is the thing the question that you most like to see answered, to see the realization of what you're talking about?
Well, this has a whole set of problems. Currently, our patients have a plug on their head. They have a cable going to a big rack of computers, and we have the mystery of actually what the brain is doing. So I have the problem of wanting to know what the brain is actually doing, not what we think it's doing. I want to be able to make a device that will be hidden inside the body, much like a cochlear implant or a cardiac pacemaker. And I want to take this big chunk of computers and shrink them down to something you might wear like an iPhone in your pocket. So those are our problems.
What question do you most want answered?
So if you come from the point of view that I do, which is to look at a really simple system, what you're seeing is emphasized are the similarities and not the differences among different diseases. So we can model different diseases in the fly, but they look more similar than different. And so our idea that we're going after is that there might be common gene networks that underlie multiple different diseases, and if there are, then it means there might be gene nodes that we could attack for therapeutics. And so we'd like to identify those gene networks that might tie multiple different diseases so that we can go after those in a therapeutic sense.
John?
Yeah, so I have a comment and a dream. The comment is I think the great message that this show can put out is that the nervous system and neurological disease and neuroscience seems to benefit greatly from multi-level explanation. We've had genes, proteins, receptors, neurotransmitters, and I think that's not just about taste, even though Eric made the very great point that psychiatrists and neurologists seem to have their own preferred level of entry into understanding. But the profound point is we need to attack all these levels and have research attacking all these levels and then having everyone interact. So that's my main comment. I think that's a very profound thing about biology versus physics, for example.
Alan?
My real question is kind of where I started this business. I was trying to understand the basal ganglia.
Right.
I do not understand the basal ganglia. I understand how they can cause all kinds of problems when they don't work, and how we can help with that, with these ways of modulating activity. But I think we really don't understand exactly what they're doing, and I think the evidence is pointing more and more towards something we talked about, which is plasticity and learning. And I think this probably will be a more important and profitable venture than trying to figure out what we've been studying to this point. And it's a big topic, but that's where I think the answers will be.
My colleague?
I would like to see the kinds of logic we've heard tonight around this table applied more extensively to psychiatry. I think psychiatry is lacking in emphasis on anatomy, on how different regions function electrophysiologically, and I think that kind of thinking needs to happen on a routine basis. And one of the things we were discussing before we came in here is to what degree ought to be a common training for neurologists and psychiatrists, at least for the first several years of their career.
Now, is it psychiatry that's not receptive to this, or what?
Some of my best friends are psychiatrists. These are cultures that have grown up over a long period of time, and it's difficult to bring them together, although the leaders understand that this convergence is necessary.
Remarkable panel. When we reconvene here in 2 months, what will we talk about?
We're going to speak about the role of the brain in decision-making. You, Charlie Rose, make a lot of decisions in the course of your life. You make personal decisions about whom you form partnerships with. You make social decisions about what clubs you want to affiliate with. You make emotional decisions of all sorts. Moreover, not only do we begin to understand that, we're beginning to understand how people make economic decisions. And this is allowing a dialogue between economists and brain scientists to see what are the mechanisms that underlie economic decision making. Do you want a short-term gain, a small gain that you can be fairly sure of, or a much larger gain that is a little bit chancier?
The larger one.
What are the kinds of strategies you're going to use? Moreover, we realize that certain lesions of the brain make you irresponsible as far as decision making is concerned. So emotion plays an important role in this, and disorders of emotion can interfere dramatically with your ability to be a rational decision maker. So we're going to consider this next time.
This is remarkable stuff. It really is.
It is
when you
wonderful
just
stuff.
think about what we're learning, you know, I mean, it's like this most amazing organ, and we're just beginning to understand it, and we find out that we're building on the history of great people who had remarkable insights without any of the tools
Absolutely right.
that we have.
This
It's
is
amazing.
the extraordinary thing. The major people like Darwin and Sherrington and Penfield, they did extraordinary work with really primitive tools. Being creative and bright helps.
And it certainly had that. Thank you, my friend. So there it is when we come back, The Deciding Brain. See you in September.